Again, another goal is to provide sufficient number of plantlets. It's the same thing because we want to produce, multiply vegetatively, and you actually overcome the problem of seeds because not all plants produce seeds or not all plants can be grown from seeds. So which is very difficult. You know, some plants, they would give rise to small plantlets, their roots, and that's the only way they reproduce. That's the only way you can propagate them because they do not have seeds or their seeds do not grow. So micropropagation is the answer because they really well respond well to the vegetative reproduction. Advantages of micropropagation would be more or less similar to what we had for plant tissue culture. So the first one is rapid and efficient plant multiplication at scale. Then the genetic and phenotype. Okay, this is very important. When we perform a plant tissue culture, there is always a chance that during the division and subdivision and the differentiation, some of the genes may alter. But this is in our control. When we work with plant micropropagation, we have a clonal multiplication. Clonal multiplication means the genetic makeup remains the same. If you have one plant, let's call that plant C. And then after micropropagation of 1 year, we have 1 million plants C. So all these plants, they will be clones of each other. They will be having exactly same genetic makeup. We will make sure during micropropagation, the genes do not alter, that there is no genetic change in these, during the tissue culture micropropagation plants to these genes, because it could be an important plant in which through a lot of hard work, we have inserted a very important gene. And if we cannot ensure that the gene will remain the same, then this is useless. So in micropropagation, we ensure that there will be a clonal multiplication and everything would be having the same genetic makeup at the end of the process. High healthy plants, because how can we ensure this? Because the TC represents tissue culture. It's a process that is sterile and disease-free because we work in the sterile and controlled lab environment. Pathogen-free testing, pathogen testing certification. This is one of the method because you can take plants and you can do testing on them if there are any pathogens in these plants. This method is also used for that purpose. Because you're working in a lab, so a few meters square, of course you need a lot of equipment, but a few meters square is enough for making thousands and hundreds of thousands of plants varieties at the same time. And there's the reduced risk of maintenance of high value stock genotypes. Why? Because we are in the lab, because we are well protected, because we are protected from, because it's a controlled environment, we are protected from viruses and fungi and bacteria. So if you have some genotypes which are high risk in these labs through micropropagation, you can vegetatively grow them under reduced risk conditions. Disadvantages, costs, of course. You cannot just work anywhere for micropropagation or tissue culture. You need a sophisticated lab. You know, there are a lot of equipment required. There's a lot of filters required. You want the air to be filtered. You want the water, the autoclaves. You need a lot of medium, growth medium. You need a lot of glassware and good quality glassware, not the soda glass, so that there will be less and less toxicity. So it will require some costs. Skilled labor. When you're working in a farm, you can have the tools for tilling. Anybody can do this with a little bit practice in the normal conventional methods. But in a tissue culture lab, it's not it's not a walk in the right for everyone. You need skilled environment, the skilled labor. So skilled labor would also costs. I mean, you need people. You have to train them. After training them, you have to hire them, and even only then they can work. Aseptic is really important, which is the next point. Losses due to infection. Because if one spore of fungi, of one fungus enters, it can really ruin the whole lab, and you would see there will be fungi growing everywhere because the conditions are ideal for plant differentiation, the in vitro condition of plant tissue culture, they are suitable. They are, how do you say, ideal for plants. But they are super ideal for bacteria and fungi. Why fungi? When in plants, the biggest threat, the biggest pathogenic threat to plants is fungi rather than bacteria and virus. Okay, there are diseases in plants which are caused by bacteria as well as viruses. For example, tobacco mosaic virus, it's a virus. Retinonecrotic mosaic virus, it's a virus. Potato virus X, potato virus Y. The blight of potato is Phytophthora infestans, it's a bacteria. Citrus canker, it is another bacteria. There are diseases in plants, vegetables, fruits, of course, everything, that are caused by viruses and bacteria. But the biggest problem the plants have is from fungi. They grow like crazy on plants. For example, Vaccinia graminestrici, Argot of Rye, a number of more diseases are caused in plants by fungi. So losses due to infections. If you do not work aseptic, there will be a problem. And you will have infection all over your lab and you would have to clean everything up. And that's not easy. That's one of the disadvantages, which you do not have in conventional farming. Micropropagation's critical factors, again, facilities. You need to have laboratory, a well-established laboratory. The tissue culture growth room, entrance of air and the entrance of people must be really controlled well so that no contaminants enter the lab. Nursery is another very critical point, because after creating plants in tissue culture, you will have to eventually transfer the plants to nursery. And the nurseries should also be well protected from all these things, because at the early stage, when you transfer these plants from tissue culture to the nurseries, to the pots, they are very fragile. So a lot of care is needed. The source, mother plants, you must ensure that it's a healthy tissue. If you bring a tissue that is already infected or that is already contaminated, you're getting into trouble, because all the cultures, all the callus cultures that you have, they will be contaminated. And this is very difficult because for surface sterilization, you can use sodium or potassium or calcium hypochlorite. You can use a lot of other like alcohol, but you cannot really see if the bacterial or viral or fungal pathogens are inside the tissues. Very important to have a healthy source. A workflow, how do you work? And the sterile media formulation, of course, optimization, it's very important. And again, skilled labor, this is really one of the first and foremost requirements.